Okay, so this is a finished part of translation here, and the end of 6.4. I also have a little practice that we're gonna do together. So this is the written component where I discuss these two pieces here that are added to the mRNA. So this is some editing. Before exiting the nucleus. Okay, and this is done to the messenger RNA. Okay, and this is what I showed you in that picture where those caps and those tails are added in order for it to make its way to the ribosome, which is going to read it in order to get our protein code. Okay, so translation. Translation, in the most simplistic terms, is taking mRNA and making a protein. Okay, those are our two steps that we're going to go through in translation. So the translation of mRNA to generate a polypeptide occurs at the ribosome. They are in the cytoplasm. We learned that there are free-floating ribosomes in the cytoplasm. We also learned about them being attached to the ER. These are not the ones attached to the ER. These are the ones specifically free-floating in the ribosome. Okay. If we're talking about prokaryotes, this process is not as complex. Prokaryotes' job is to make more cells and make them quickly. So transcription and translation inside prokaryotes like bacteria is going to happen seamlessly. It's going to happen quick, it's going to happen efficiently, and since they only have that small circular DNA, let's take a look at that again. This piece right here, transcribing and translating of this is going to happen a lot faster than our huge DNA double helix. Okay. So I'm going to show you a couple pictures in a second, but translation is going to involve quite a few steps. We're going to talk about attaching it, the building of the protein, and releasing the protein. We're also going to talk about this specific word here, codons. Codons are the language that we're reading. Those A's and U's and C's and G, those base pairs are going to be read in a specific way in order to give us a molecule that we're looking for, okay? You learned how to do this in freshman bio. I'm just giving you a quick reminder about it. Okay, so those codons are going to specifically be on the mRNA. The mRNA has the code that we're looking for. Each codon encodes for a specific amino acid. I'm going to teach you how to read the codon wheel and know specifically what's coded for and what amino acids you can find using what codes. And then just a couple things in terms of that codon wheel. Some code for one, some code for more than one. There's a little variety there. There are 64 combinations with 20 amino acids. Okay, and if you're like, I don't remember what amino acids are, I don't know what you're talking about just yet, I'm going to show you on this wheel and clarify it for you. It's very easy to read. It's something you can pick up in just a couple minutes, okay? So let's take a look at translation. Okay, so translation is going to begin with a specific codon. The codon is AUG. AUG is going to be our start codon. Okay. Here is your ribosome. When the mRNA or when the ribosome reads to the mRNA, it's not going to start making a protein until it gets to this codon, AUG. Okay. And they're reading this codon in 3 bases. Every 3 bases are going to code for a specific amino acid. So before I get into tRNA bringing the amino acid, let's take a look at the codon wheel. Okay, here is the codon wheel. The codon wheel has all the combinations that code for all of your 20 amino acids. The 20 amino acids that are ones you get from your diet or the ones that your body makes specifically for you, okay? But these are integral to making more proteins in your body. On the codon wheel, AUG we said is our start codon. So the way you read this, you start in the center with the first letter of the codon. The next letter down is smaller, and the third letter you find in the outer perimeter of the wheel. So for a start codon, we have AUG. You might read this as methionine and say, hey, Miss B, that doesn't say start. Well, methionine, since it's the only combination that you get for this specific amino acid, this is our start codon. Methionine and start are synonymous with one another. Okay. There's a couple other things I wanted to point out on this wheel as well. There are 3 combinations for a stop. What you're going to notice when we build a protein, the ribosome is going to read, it's going to start at a start codon, it's going to add amino bases until you get the specific protein sequence you want, and then it's going to reach a stop codon. And there are 3 of those. Okay, so one of the stop codons you can find here. It is UAA, so we have UAA, UAG, UAG, and UGA. Okay, those are your 3 stop codons. Now the other combinations here, you may see that alanine has 4 combinations of codons to code for this specific amino acid. Valine, same thing. Arginine only has 2. If we're talking about tryptophan, tryptophan has 1. But how you read this wheel, these are specifically for the codons on the mRNA sequence. Okay, you read mRNA, this is the tool that you need in order to code what specific amino acids are making the protein that you have in front of you. Okay, so back to our picture. Here's our ribosome reading our specific mRNA. It's going to find these codons, and then it's going to initiate tRNA to bring something called the anticodon. The anticodon is what matches, in parentheses, matches the codon that's on mRNA. They're going to fit together with the complementary base pairs in RNA. RNA now has these rules. RNA, A pairs with U, C still pairs with G. And you can see here, in order to drop off our start codon, AUG is going to pair with UAC. Those 2 pair together using those complementary base pairs, and it drops off the amino acid. Okay, and that's going to continue as the ribosome reads through the specific mRNA sequence, codon after codon. Now, if we look here, PHE codes for phenylalanine. One thing I want to point out, we are not reading this anticodon, we are reading the codon here. So if I go to my codon wheel, UUU, which is the code here,